All right, class. So looking into the mechanism of urine formation. There are four main mechanisms that help in the process of urine formation. The first one is glomerular filtration, and we did mention that this process takes place in the glomerulus only. The next process is selective reabsorption, which takes place in most part of the tubule. And we also have a special mechanism called tubular secretion. And then finally, all the filtrate goes into the collecting duct, and the collecting duct excretes the urine out of the kidney via the ureter. So, glomerular filtration. So here I have drawn out a blood vessel, interstitial space, and a nephron tubule. I want you to notice that the blood vessel is lined with simple squamous endothelial cells. The interstitial space is the space that all these materials that pass the blood vessel or that come out of the nephron tube, they have to pass the interstitial space, right? So things will move from the blood vessel into the interstitial space into the nephron tubule, or the other way, from the nephron tubule into the interstitial space into the blood vessel. Got it? Okay. So I'll start glomerular filtration in color, let's say yellow. So glomerular filtration, things from the blood vessel go to the nephron tubule. This is called glomerular filtration. Where does it take place? Takes place in the glomerulus only. Glomerulus only. And remember, this is a bulk process. So a lot of things are actually filtered out of the glomerulus into the Bowman's capsule. So things get filtered out of the glomerulus into the Bowman's capsule. And by this process, a lot of stuff gets filtered out. For example, a lot of water gets out. Glucose gets out. Salt gets out. Vitamins. Amino acids. Urea. Creatinine. Uric acid. All these stuff gets filtered out of the blood and gets into the nephron tubule. However, if you look at, and this soup thingy, this is called filtrate. That's called filtrate. Hope that's clear. Now the problem is, now we have filtrate in the Bowman's capsule. But look what is there in the filtrate. We have glucose, we have amino acids, we have vitamins, we have salt. Obviously, we cannot get rid of all those important stuff, right? So at other part of the nephron tubule, like the proximal convoluted tubule to be more specific, that is a site where a lot of these things will be reabsorbed back into the peritubular capillary. Make sense? So let me see. I'll use a different color. So let's say at the level of the proximal convoluted tubule, a lot of these stuff will move back into peritubular capillaries, right? And this process is called selective reabsorption. Reabsorption. So that's that. Okay. Now, selective reabsorption also takes place from the nephron loop to the peritubular capillaries and vasa recta. Then there are specific areas of the nephron tubule called distal convoluted tubule. It is at the distal convoluted tubule, and also actually other parts of nephron tubule we have the same thing going on. But tubular secretion is a process where specific things are dumped out into the nephron tubule. So if you have a specific thing, for example, let's say you have these specific molecules that you want to get rid of. So these specific molecules will move into the nephron tubule by a special process called tubular secretion. Now, Rupa, so what's the difference between glomerular filtration and tubular secretion? Well, first, glomerular filtration only take place at the glomerulus, and it's the filtration from the glomerulus into the Bowman's capsule. Glomerular filtration is a bulk process, so a lot of stuff gets filtered out of the blood into the filtrate. In case of tubular secretion, it takes place at other parts of the nephron apart from the glomerulus, and by this process only selective things move from the blood vessel into the specific part of the nephron tubule. And then the fourth process is called excretion, which takes place from the nephron tubule out into the ureter. So that's excretion. All right. So we have talked about all these processes that lead to urine formation. All right. Now we will look into the renal corpuscle. What is that? Okay. So let me draw this thing one more time. So you have the afferent arteriole, you have the efferent arteriole, you have the glomerulus. Surrounding the glomerulus, you have the Bowman's capsule or glomerular capsule, leading to the proximal convoluted tubule. This thing is the Bowman's capsule, also known as glomerular capsule. Cool. What else do we see? We see the afferent, actually I should label it with a different color. We see the afferent arteriole. We see the glomerulus. We also see the efferent arteriole. Now listen carefully. The Bowman's capsule plus the glomerulus forms the renal corpuscle. So renal corpuscle equals to Bowman's capsule plus glomerulus. Together, these two things together form the renal corpuscle. Very, very important. Okay, now let's look into the Bowman's capsule a little bit more in depth. Know that the Bowman's capsule is lined by epithelial cells. The outer layer of epithelial cells, this layer that I'm marking with green, this is simple squamous epithelium. Simple squamous. So this is simple squamous epithelium, and these are called, this layer is called the parietal layer. We will see a very interesting visceral layer. The visceral layer of cells of the Bowman's capsule looks very different, and these cells are called, I should draw with a different color. These cells, they are these ugly cells, and these cells are called, I'm drawing them in blue, these cells are called podocytes. What are they called? Podocytes. Okay, all right. So we are looking into the Bowman's capsule. The parietal layer is made up of simple squamous. The visceral layer is made up of specialized epithelial cells called podocytes. Now, make a fist. Make a fist. Imagine the fist is the glomerulus. And then with your other hand, try to cover the glomerulus or try to cover the fist. So your other hand that you are using to cover the glomerulus, though you can imagine those as podocytes. Does it make sense? So I want you to understand that podocytes, they actually cling to the surface of the glomerulus. They cling to the capillary network of the glomerulus. And we will take a look into a picture from your textbook a little later. But before that, I want to focus on a very, very important concept. I will blow up a podocyte. Ew, this ugly looking cell is a podocyte. All right. And then I'll draw out another podocyte. Let's do this. This is another podocyte. What I want you to understand is that this whole cell is a podocyte. And these extensions, these finger-like extensions, these are called pedicels. These are called pedicels. I want you to understand that the pedicels interdigitate. Do you see this area? Do you see this part? Actually, I should highlight with a different color. Do you see this part and this part? Do you see how they're interdigitating? So the pedicels interdigitate, digitate. Really important. So they interdigitate, and in between them you have the glomerulus, right? Red. In between you have the glomerulus here. That's the glomerulus in between. The point is, this interdigitation helps in the filtration process. Another thing that I want to point out to you is that if you look at the glomerular capillaries, these capillaries have holes. They are called fenestrated capillaries. So these are fenestrated capillaries of the glomerulus. Okay, so what's the key point here? What's the take-home message here? We do know that the glomerulus has fenestrated capillaries, meaning these capillaries have pores through which larger materials can pass out. The visceral layer of the Bowman's capsule is made up of special cells called podocytes, which interdigitate by the pedicels, and together the porous capillary of the glomerulus and the interdigitated podocytes, together it forms something called filtration membrane. What is it called? Filtration membrane. Man, my handwriting. Oh, what? Did everything get deleted? No. I'll cross this out. This is called filtration membrane. So again, one more time, filtration membrane is formed by the fenestrated capillaries of the glomerulus and the interdigitated podocytes. Together they form filtration membrane, across which filtration takes place. Okay. All right, so this is a picture from the textbook. What do we see here? We see the afferent arteriole. We also see the efferent arteriole. This thingy is the glomerulus as labeled here. Glomerulus. Now I want you to pay attention to the Bowman's capsule. The parietal layer of Bowman's capsule, this is the parietal layer of glomerular capsule or Bowman's capsule. This is made up of simple squamous epithelium. Now let's look into the visceral layer of cells. The visceral layer of the Bowman's capsule is made up of podocytes, which are these orange octopus-looking cells. Do you see that? And these podocytes, they cling to the surface of the glomerular capillaries, right? And they interdigitate. Do you also see them interdigitating? Yes. And together that helps to form a strong filtration membrane. Remember, the glomerular capillaries are also fenestrated. Then this part is a tubular pole, and that leads to the proximal convoluted tubule. Hope this is clear. I'll start with a